Now.
Good evening, everyone. Thank you very much for joining us. My name is Trevor Young, and together with Helen Moran, we're your co-chairs for this evening's webinar. It takes me great pleasure to introduce our first speaker. That's Matt Lee. He is NIHR Clinical Lecturer in General Surgery at the University of Sheffield. Thank you very much, Matt. Over to you.
Lovely, thank you very much. Nice moment there of almost failing to unmute myself. So good evening, everyone. My name is Matt Lee. I'm a general surgery lecturer, researcher at the University of Sheffield. It's really my pleasure to be here talking to you tonight or this evening or wherever you are about systematic reviews and how to some tips for how you can do a good systematic review. I have. Make sure I can move my slides. I have no conflicts of interest to declare. What I'm hoping to achieve here is quite a high level review. We've got limited time and I could go for days on the topic. So I'm having to work from the point that people know what a systematic review is and have either had a go at one or have read a couple and want to learn a bit more about the nuts and bolts. It's going to provide an overview of systematic reviews, really focusing on how to prepare and develop a question and talk a little bit about the steps of doing a review, along with some sort of top tips or key thoughts as we go along. A lot of these come from my experience of doing systematic reviews and of being a peer reviewer where, unfortunately, I often end up being reviewer too for these. So hopefully it'll be useful. So the first question is, what is a systematic review? It's actually can be quite difficult to pin people down on what it is. They can look at something and tell you whether it's a systematic review or not, but the definition is a bit unclear. And this is probably the best definition that I found from a 2019 paper, which was a systematic review to define systematic reviews. These guys say that a systematic review is one of those reviews of the literature that reports or includes a research question, a clearly defined research question. It can tell you where people searched for to find papers or information, and it provides a reproducible search strategies to tell you where they looked and how they looked and when they looked. It tells you how they selected papers that went into the systematic review. It's a clear inclusion and exclusion criteria and very clear selection and screening methods. So how did they go from a big pile of papers to, the final list of 20 or 30 included papers? Also importantly, critically appraises and reports the quality and risk of bias of the included studies, and I'll get onto that later, why I think that's one of the key things in a systematic review. And finally, it should tell you about how it handled data, particularly numerical data in the form of meta-analyses. I'm not going to touch. That's going to be for Dr. Fleming to talk about shortly. So that's what a systematic review is. Slightly academic definition. So what does it mean in practice? Well, if you're going to do a systematic review, it means you spend some time thinking about how you're going to do it and writing down a question. You then do some searches in some databases using a search strategy. It gives you a massive pile of papers, which might be in the hundreds to the thousands. You then, along with another reviewer, go through that big pile of abstracts. You don't read the full papers, just the 250-word abstracts, and decide whether this paper is in, out, or a maybe. You then retrieve the ones that are definitely in or maybes and look at the full texts and decide whether they're in or not. And then you and your co-reviewers will extract the relevant data, do a bias assessment and write a paper. It's quite straightforward, isn't it? I think the average, whilst it sounds like it's quite straightforward, and systematic review is often used as a sort of gateway to surgical research because it's viewed as being an easy thing to do. It's worth pointing out that the average time for a systematic review to be done is heading towards, I think, about 12 months. It's doable much faster, but that's only if you've got dedicated time. So there we go. So that's what most of you will be familiar with. And I could end the talk there, but there's things that we need to dig into and talk about in more depth. So why are we going to do a systematic review? I think there are three main reasons why we do systematic review. One is that we think there's enough evidence out there to tell us how we should change our practice. So that we could talk about the roles of neoadjuvant chemotherapy in preventing recurrence after after surgery for colorectal cancer, or preventing a systemic disease after colorectal surgery. That might be something that we can do a big review on and synthesize data that tells us, yeah, this is worth doing, or no, it's not worth doing. Another reason you might do systematic review is to is that you know that there's an intervention that offers benefit, but you need to get an idea of how people are doing it. And so you can review that and look at the different steps that are involved. And that might be if you're looking at how people offer a laparoscopic resection, how many ports they're using, what size ports they're using, where are they placing them, what energy devices they're using, if any, all these things that you can pull out of a systematic review and a big data set. Or finally, it can identify questions or gaps in the literature. And sometimes you could do a systematic review and not identify any papers that answer your question because no one has dealt with it. Or even better, you might find some papers that give you such varying or wild results that you know there is a clear question to be answered, and that can help you inform your next steps in your research. So the first thing we need to talk about is planning your review. A lot of people try and rush to doing the searches and everything else, but you have to pause and take a breath. And there's that old adage of failing to plan is planning to fail. These could be, as I said, they're viewed as being an easy way of doing research, but actually they're quite a complicated and complex method when you start thinking about things, or if you stop to think about the nuts and bolts behind the method. I would recommend that anyone who's thinking about doing a systematic review at least bookmarks the web page for the Cochrane Handbook of Systematic Reviews. It's freely available, it is comprehensive, and it is essentially it's a holy manuscript for anyone who's going to do a systematic review. It has some sections which are easier to read and bullet pointed, and some very high level summaries, and I think you should at least have a think about the summaries on different aspects of a review before you jump into it feet first. It'll save you problems later. Next thing you might want to do is, if you've got an idea, so let's say you want to do a piece of work looking at minimally invasive surgery in rectal cancers, you might want to know if someone else has done a systematic review on it before. And the first place I would go is to the Prospero Registry of Systematic Reviews, and that's free to access register that's hosted by the University of York on behalf of the National Institute for Health Research. And people are expected to prospectively register their reviews into this, and they'll tell you where they're going to look. They'll tell you what their PICO, what their research question is, what databases they're planning to look in, what search strategy they're planning to use. So they'll give you that list of all the search terms they're planning to use, inclusion and exclusion criteria, and all the other assessments that go with it. And that's quite a nice. I find these really useful things to look at when I'm thinking about doing a review, because it triggers thoughts on, oh, actually, I haven't thought about how I deal with that situation if it arises, or actually maybe there's an exclusion criteria I haven't thought about. It does aid for transparency, and that's one of the things that definition of systematic reviews talks about. And it does just make you think. So I would recommend looking here, and also the Cochrane database of systematic reviews if you've not already looked there for an example review. And finally, this is something you're not going to use until the end of your review, but the PRISMA guidance is something I'd strongly recommend people look at. This is a set of guidelines for reporting a paper, so essentially tells you what headings you need to have in your paper and what you need to cover in it. It's freely available to access. The checklist is about 2 pages long, and the supporting documentation is easy to read. You should really spend some time thinking about the Cochrane Handbook, about having a look on a register of reviews, and also on PRISMA before you really get started. So having looked at those things, at least in passing, you're going to think about defining your research question. I know that there's a recent webinar where this was dealt with, so I won't go into too depth, but you need to come up with a PICO question, or which is either population intervention, comparator outcome, or population exposure, comparator outcome. And it is formulaic, and that's because it informs how we structure our search strategy. So obviously the question that you're forming is, in a population, does the intervention or exposure compared to the comparator or non-exposure affect your outcome? And that outcome is your primary outcome, the outcome of interest that all your papers are going to report on. So for example, an example of a PICO, and we can debate how good this is, in adults with a low rectal cancer, does laparoscopic surgery compared to robotic surgery affects local recurrence? That's an example of a PICO. And some of you will be sat there thinking, actually, there's other things that I'd be considering here. And that's good, because that's where we're going with the next slide. There are other things you might want to think about, particularly in surgical oncology. The field is, constantly moving. you may want to think about limiting your searches to a time period, maybe the last 20 years. And certainly laparoscopic surgery was developing during the 90s, and it's become a lot more mainstream since the year 2000. So you want people, if you're going to look at laparoscopic surgery or minimally invasive surgery, you want to be looking at people who are well up their learning curve. You might be doing this on the cheap, so you have no funding available. Are you going to be able to include studies that are not in English, or not in English plus another language? Quite often you'll find that there are some niche European journals that have a paper you might be interested in, but they're not in English, and there's quite a few journals that publish literature in Chinese. So you can struggle to, if you don't have a native speaker and you don't have funds, you might struggle to include them. Are you going to go out and try and get all these papers? You need to be clear on the kind of studies you want to include. So if you're comparing two interventions, do you really want to have a case series of 10 patients where the two things have been used? Is that going to add to your findings? Does a case series of 20 patients, 30 patients become useful? When are you going to be interested in retrospective cohort studies, prospective cohort studies, randomized trials? You need to be clear on the kind of studies you want in, and if you are including or excluding a design, you need to be clear why you're making that decision, and you need to be able to justify that to the reviewers. So obviously randomized trials reduce a lot of the elements of bias and treatment allocation bias. Perhaps you want longer term outcome data, which you're only going to get through a cohort study. It's things to think about. Are you going to exclude specific comorbidities or specific other treatments? So for example, if you're looking at quality of anastomotic leakage, or quality of or risk of local recurrence, you might want to exclude people who've had radiotherapy to their pelvis in rectal cancer, given that this is people who might have been higher risk of having an involved resection margin, or might be high risk of that local recurrence. There's things that you might want to think about there and just tweaking your inclusion exclusion criteria. And when you've been through and really defined beyond that PICO, that list of extra things, that's where you've got your PICO and your inclusion and exclusion criteria. And a lot of people get partway through the search and will then go, you know what, I really wish we should have X as an inclusion or exclusion criteria. It's too late then. You've got to do it now to be robust and reproducible later. Next thing you've got to do is get the searches right. And certainly as a reviewer, this is one of the things that I often pick up on, because there's a form of words you expect to see, and where you don't see that form of words, you pay a bit more attention. So where can you look for information? And there's a few databases that we have, one of which is people will be familiar with, it's Medline. That's by the NLM, the National Library of Medicine, not the NIH. And basically indexes all medical journals, what we class as a medical or surgical journal. Also Embase, which is managed by one of the major publishers, and that's again another index for all the major medical journals. There's a lot of overlap between Medline and Embase, but they have different search functions, and you can run a very similar search in the two and get different papers out. So it's often worth searching both of those. CINAL is another database. It covers nursing and allied health literature. So if you're doing some work looking at physiotherapy, physiotherapy interventions, nutrition interventions, nursing interventions, and particularly wound management interventions, that's going to be in CINAL. So you're going to have to go and search that database. PsycInfo is maintained by the American Psychiatry Association or American Psychology Association, and it tends to focus on the psychology and psychiatry literature rather than that medical literature. So again, if you're looking for qualitative work, you're looking for psychology-based work, you're going to have to include this. A lot of time people will talk about the central database. It's managed by Cochrane, and it provides sort of short summaries or indexes summaries of randomized trials or quasi-randomized trials that have been published in the literature. So particularly if you're doing a systematic review that's focusing on randomized trials, you need to be going to central and having a look there. And the other big source we talk about is the grey literature. And what we mean by grey literature is stuff that we know is there, but isn't necessarily indexed in one of these sort of robust and reproducible databases. So it might be the unindexed abstract, the abstract from a conference that's not been indexed, not been published in an indexed journal, it's just online in a PDF, or a policy document from a government or a society. It's generally that stuff that you find through internet searching. And that can also be very important. When you're looking for information, the way you look for information is also important. I'm going to talk about search strategies in a second, but the way you access these databases is important. Most of the time we use something called OVID, which is a portal to an interface into these databases, and that's got a reliable and replicable syntax, as in the structure of how it asks for words and the functions that you can apply to those words to get things, and that's reproducible. A lot of you will know that you can search Medline using the PubMed interface. PubMed isn't a database in the way that these other things are. It's kind of a search engine that looks at Medline and a few other things. PubMed also does, basically is clever like Google in that it will automatically expand your search to other relevant terms, and so it's not quite as doesn't give you quite as reproducible results as searching Medline through the alternate interface. And a top tip is, if you say in a review, we looked at two databases, we searched PubMed and Medline, I'm going to call you out because you've essentially searched Medline twice, and that's a bit of a waste of your time. So when you're designing that search strategy, you need to have the key terms to put into it. How are you going to find these key terms? Well, you're going to look at search strategies from similar reviews. That's where Prospero comes in. It's where the Cochrane repository comes in. That'll help you narrow down things related to your disease strategies, and help you get an idea about how some of the operators work. You might also know the paper, you might have an example of a paper that you expect to be in your review, and you can go look that up on PubMed and look at the MESH terms, the medical subject header terms, and include those in your disease searches to make it a bit easier. You need to get yourself familiar with how each of the search engines work, because they're all a little bit different, and there are online tutorials for some of those. When you're searching the grey literature, so that's going through your abstracts and things, I'd recommend using an incognito or cookie-free browser, because Google will try and show you what it thinks you want to see rather than what someone would see on a blank search. This can be quite complicated, and some of you are probably sitting there thinking, this sounds like it could be tough, and it can be. You've got to get it right, otherwise your review's going to be rubbish. So most institutions, that's hospitals, universities, have a librarian who'd be interested in helping you out, helping you refine your search strategy, or getting access to the databases. And finally, I'd recommend doing a test run of your planning to do a test run of your search strategy, and making sure that those key references, the papers that you expect to be included, are indexed. If they're not, you've done something wrong with your search strategy. As we said, it's got to be replicable. So one of my top tips with it will help you with writing up and getting past peer reviewers, is that when you've run your search, you can copy and paste it into a Word document, state which database it was used for and the date of the search. When you've done that, it's an appendix for your publication, and you've dealt with one of the most frequent things that I pick up when peer reviewing a systematic review. You've got to get your data management right. This is, when talking about systematic reviews might be considered quite a dry thing, and data management equally is exciting for a Tuesday evening. The data management here is about deduplicating your results. You've got thousands potentially of hits, and you've got to make sure that you're only looking at each one once, and I guarantee you there will be abstracts that appear twice in the search, so you've got to get rid of them. Your other things are, you've got to manage two people screening these abstracts and deciding whether they're in or out, and getting the full text and assessing and deciding whether they're in or out, and then collecting often many data points from them and making sure that the data points are the same. So there's a lot of data management that goes on here, and you've got to have the right tools for it. So when you've done your searches, you can export them as specific file types, so XML file types or RIS file types, one of which is readable by spreadsheet programs, one of which is readable by reference managers. You've got to upload it to a relevant tool. You can use a generic spreadsheet program, has its benefits, it's offline, it's local, you can go through it. You can keep everything in here and move things through onto different sheets for data extraction and other things. You could use a reference manager, which has its pros and cons. You can be quite slow if you've got lots and lots of things involved, and your filing can sometimes become difficult, but I've definitely used EndNote to do a systematic review before. More recently, I've been looking at online tools, however, things that allow me to work with other people in real time, so I can see how many abstracts my colleagues have screened, or I can see how many things we think are going through, so it gives me some real feedback on what's going on. Now there's Rayyan, R-A-Y-Y-A-N, which is made by Qatari Group. It's free to access, and I think it's quite a neat piece of software. It's very good at identifying duplicates to help you deduplicate. It's good at identifying where there's conflicts and managing multiple reviewers. I think that's a nice piece of software to use, and it's particularly good when you can see how many abstracts people have screened each. It's good for competition between surgeons. Covidence is actually a very nice piece of software, and it's made in collaboration with the Cochrane Group. The downside is it does cost money. If you have the money, or if your department has a subscription, definitely use Covidence. It makes life easy, and it also generates your Prisma charts for you, and you can do everything through to data collection into it. So I would definitely look for an online tool. So Rate and Covidence would be my preferred choices if you've got the resources. Other things you need to do: make sure you've logged the numbers at each stage, what you've got rid of where and why you've got rid of it, and managing where your full texts are. And this can generally be done in a spreadsheet. And then your final report or the final data forms you're going to have are going to list those key things that you've extracted. And in a spreadsheet, it's the easiest way to go, I think. That should be things like the year published, the first author's surname, the number of included patients, number in each arm, primary outcomes, secondary outcomes, and anything else that's of interest. Bias assessment is really, really, really important. It's often relegated to being a sentence or two at the end of the results section, saying things like, for our a bias assessment was conducted using this tool and it's presented in figure 4 or in appendix 3. That's not really helpful. Why isn't it helpful? When you are doing a systematic review, you are essentially, for the reader, we are delegating our some of our critical thinking to you. We expect you to look at these papers and decide whether they're any good, because a systematic review is only as good as the papers upon which it's based. If you put rubbish papers in, you'll do a low quality systematic review. If you make sure you get high quality papers with high quality extraction, it'll be good. And the bias assessment is where you tell us that you've thought about these things. All the papers have, structural or implicit error that's there, and you've got to tell us you've thought about it. And if things are consistent across papers, that's interesting. That means that we are getting something wrong as a group of researchers, or the literature. We need to think about a new way of doing things. Whether that's in the definition of an outcome, it might not have been defined, or we might have a weird definition, or we might have, it might be that the patients were putting in a really inconsistent, so treatment allocation is an interesting issue. For me, it's the most important, most interesting part of the review, and I like to see it in the second or third paragraph, which is where Prisma recommends it should be. You should have 2 people doing this assessment and discussing it. That's how you flesh out that paragraph, and a third reviewer engage with it. Yeah, it should be more than 2 sentences in your paper. You do this in a structured way. There's lots of tools you can use. The Cochrane risk of bias tool is that 6-domain one that you see in lots of studies. I really like it. It's good for. It's designed for RCTs. It's what I'd recommend you use if you're doing RCTs. If you've got having cohorts or non-randomized studies, the Cochrane Robbins I or the Robbins NRI is a good tool to use. It's got a few more domains. It requires a bit more thought. Some of you may have done systematic reviews and used the Newcastle Ottawa scale. It's got, I can't remember, 15, 20 items on it, and they have varying levels of assessment. And that one does, I think, have some issues with inter-rater reliability. So, and I don't find it that useful as a reviewer or a reader when I'm looking at it. It doesn't mean as much to me as some of the other tools. So use it, but just think about whether there's another tool that does the job better. And then if you're doing a review looking for prognostic factors or diagnostic tools, then there's a bias tool for everything. I used to have to look for it. And most of these will be found on there's website repositories that'll give you links to all of these. As I said, there's some issues with getting 2 raters to agree with bias assessments in risk of bias. So you shouldn't let the most junior person do this. It should be someone who understands the science, who understands the clinical problem, who understands the method. Because otherwise you'll end up with really unreliable bias assessments, or biased bias assessments, which helps no one. That's kind of all I wanted. That's all I'm going to say here before I hand over to Christina. In summary, a good systematic review needs to be transparent and replicable. So you've got to make sure everything is reported in that study. You've got to make sure that the guidelines and registries are your friends. They're not things to just say, oh, we did it with reference to this checklist, and then submit that as an appendix. Actually use them and make sure that they really add value to what's there. Because if you don't use them, I'm going to pick up on it in review and send you away to use them. So you're just going to put in the work till later. Make a clear plan with your team. Have we got the question right? Have we thought about all the inclusion exclusion criteria? Who's going to do the screening? Who's going to do the full text retrieval? Who's going to do the bias assessment? Have a plan and make sure you know who's doing what. Make sure your data management plan's clear what we're going to do with conflicts, inclusion exclusion criteria, and leave that time for bias assessment. Just as a slightly shameless plug, I do have a research-based blog at surgicalresearch.co.uk that expands on these thoughts a little bit, particularly around systematic review. That's free to access if anyone wants to use it. So thank you very much to the chairs. I think I've said enough. Thank you very much.
Thank you very much, Matt. That was very informative as always. And it's now my pleasure to introduce Dr. Christina Fleming, who's recently been appointed as a consultant colorectal surgeon at Limerick University Hospital in Ireland, and is currently on fellowship in Bordeaux. She's the chair of the Irish Surgical Research Collaborative and a research powerhouse who's going to give us some practical tips on performing a meta-analysis.
Excellent. Thank you, Helen, for that really kind introduction, and thank you for inviting me to speak tonight as well. So to get started, I don't have any conflict of interest related to the topic, but I think it's really important to state that I'm not a statistician. I am a surgeon scientist, and while I do have some basic biostatistics training, I learned a lot of the methodology that I will describe tonight through experience and working with other statisticians in formally designing and delivering a number of systematic reviews and meta-analysis over the past number of years throughout my training. So just to outline a few of the things I will touch on tonight, I don't want there to be too much overlap with what Matt has discussed already, but really what I'm going to discuss is the data and how do we quantitatively look at the data from the point of view of meta-analysis. I'll describe Review Manager, which is the statistical software that I prefer to use and would have the most experience with using in delivering meta-analyses, the different types of techniques and methodologies used, how to properly data extract and clean your data before analysis, and then some specific statistical concepts relating to meta-analysis, including heterogeneity, effects models, forest plots, and funnel plots. So all of this sounds really exciting, I'm sure. So to start off, what is a meta-analysis? So the formal definition is that a meta-analysis is a statistical synthesis of data from separate but comparable studies, leading to a quantitative summary of the pooled results. And when we look at the hierarchy and levels of evidence, a purist would describe a meta-analysis as a synthesis of randomized controlled trials. But if we look at the literature, we will see, and I have also published personally, meta-analyses not just of randomized controlled trials, but of cohort studies and non-randomized controlled trials. And we have to be cognizant when we're considering levels of evidence of where those meta-analysis actually slot in. And Matt referenced that already in his discussion on, the quality you get out at the other end of your review is really based on the quality that comes in at the primary end with regards to the quality of the studies that are performed. So when deciding to perform your meta-analysis, the first thing I would strongly recommend is to speak with a statistician or an experienced researcher who knows how to design the statistical methodology to perform a meta-analysis or have a lot of experience in performing it before. Based on that, you will decide on what software you're going to use. And it's really important that you decide this at the beginning, because it will really inform how you actually extract and manage your data that you take out of your paper, which I will describe a little bit more further on. Personally, I use Review Manager. It's a freely available software package from Cochrane Review. I think it has a lot of benefits. It allows you to perform very basic analysis. It's very user-friendly, but it does also have some limitations in, you don't have the ability to look at any major big data or omics data, or also I think a significant limitation of Review Manager is its inability to perform network meta-analyses. So when we talk about the types or techniques of meta-analysis that can be performed, the most common one we really talk about is described as direct pairwise meta-analysis. And in basic terms, what this means is that you're describing two ways of performing something. So, I'm also a colorectal surgeon, so I will talk a lot in terms of colorectal surgery, my examples. But an example here would be comparing laparoscopic to robotic colorectal resections. And the data within which you will compare this will either be continuous data, categorical data, or time to event. Then further ways of looking at meta-analysis techniques are, can we pool data from studies to look at diagnostic test accuracy? For example, if we have a number of studies that are reporting on the accuracy of MRI for predicting complete pathological response in rectal cancer. How can we combine their outcomes to look at the overall accuracy of MRI? And more emerging techniques include network meta-analysis, which would compare more than 2 comparators. So as we said already, in pairwise, it's like comparing laparoscopic and robotic, whereas with the network meta-analysis, you can also compare in that model open and transanal surgery as an example. And then the final type that I will discuss tonight is looking at big data. And this is really emerging and I think a really exciting area to look at going forward as well. So as I said, the most common way of looking of performing a meta-analysis that you will do or you will probably read about is direct pairwise meta-analysis. So if we were to consider this in the context of surgery, there are 3 types of variables that you're going to look at, either categorical, continuous, or time to event. And examples of such would be for categorical, what complications, is there a complication, yes or no, anastomotic leak, present or absent, continuous variables, things like blood loss, operative time. And then time to event examples would look at recurrence and survival outcomes relating to cancer. So these are actually probably the most important slides, and it's the data management, because particularly if you're starting off and you're looking for advice on how to do a meta-analysis, the most important thing to do is to get your data extraction correct from the beginning. And as Matt also described in his talk about having 2 reviewers performing the process independently, and then having a 3rd reviewer who will, I suppose, have a discussion amongst the 3 of you, should there be any discrepancies or confusion about what should be included or not. And this is really important for data extraction as well. It's important from the beginning to decide what data you're actually going to extract, because it's going to take a lot of time to extract the data from multiple papers. So you need to be really sure that what you're extracting is actually is relevant to your paper. And for example, when you go to publish your paper, the reviewer isn't coming back asking you, well, what was the average BMI amongst the group, because that might be really relevant to your research question. So sitting down as a team at the beginning with the entire authorship of the paper, but also with your statistician or the person who's going to be managing the data is really, really important. And this is something that can be performed in Excel quite easily, and most of the software packages for performing meta-analysis will allow you to import your Excel files as well into the formal analysis down the line. So here is an example of the I suppose, initial clinical data that you might consider when you're including in a paper. So this is a paper that we published looking at functional outcomes after rectal cancer and whether patients had surgery performed laparoscopically or robotically. So it was obviously really important to know when the papers were published, because that can impact on, learning curves and the integration of robotic surgery. So that's really important to consider when considering the overall outcome. It's important to consider what numbers were included in each study. It's important to consider whether patients had neoadjuvant therapy or not when considering functional outcomes, what level the tumor was at, and then finally looking at, well, how actually what were functional outcomes measured. And then when you come down to the actual meta-analysis data, so this is going to be the numerical data that you input to your meta-analysis software to generate your forest plots at the very end, so your actual synthesis of your qualitative data. And it's really important before you start to do this that you speak with the person who will analyze the data if you are not that person, because the type of data you need to extract from the paper will very much depend on the software package that you're going to use. So for example, in the first box here, this is some unpublished data that we're working on at the moment. And this is looking at operative time between 2 surgical techniques. So operative time is obviously continuous data. And in the software package that we use, the software wants to know, well, what is the mean, what is the standard deviation, and what are the total number of patients within each comparative group within each paper. So having that blank table ready for the data extractors to complete is really important, because if you look beneath that in this same study where there is an outcome of conversion, which is the categorical variable, and we're looking at was a conversion present or not, yes or no? Well, here all we need is the N value for conversions and the N value for total patients. So again, having that blank table ready to fill in for the data extractors means that they're not collecting the incorrect data across the two settings, and that will really help kind of streamline the process, but also ensure the accuracy of the data as well, if only the relevant data is being extracted. Now, in some settings, you may have a median and an interquartile range, but the actual package wants to know what is the mean or what is the standard deviation from the point of view of its calculation. So there are a number of ways that are, robustly referenced in the statistical journals on how to manually calculate mean, standard deviation, or convert odds ratios to median and quartile ranges. And the website that I have here allows you to do that online, and it's free to use, and really, really useful, because invariably, a lot of the time, this will be something that you have to deal with. And if all else fails, I would also suggest contacting the authors of the paper, as they will have the raw data and might be able to provide the data you require. And generally, they're quite willing to do that, as it means that their paper is going to be cited within a review. So one concept, two concepts, I guess, to talk about before we get into the forest plots, because these will be present in forest plots. So just to get an understanding of what they actually mean before we get on to that. So the first thing that you probably hear about a lot in meta-analysis is heterogeneity. So heterogeneity in study design, but also heterogeneity in the data. And the Cochrane-Q statistic is the most robust way of reporting on heterogeneity in studies. And essentially what this is it is a relative reporting of the variability of the data and the variability of the outcome based on the weight of the data within the pooled analysis. A far more basic way, and I would say more utilized way of reporting heterogeneity, is using what we call the I2 statistic. Your forest plot will generate this for you. And it is generally classified, if the heterogeneity is low, it's probably under 50%, and if it's high, it's over 50%. And this becomes important when you are selecting what model of analysis you're going to use, because if your heterogeneity is low, that assumes that all subjects are similar and all interventions were similar within the study design. So for example, if you're doing a meta-analysis with RCTs that have very strict inclusion-exclusion criteria, then you probably find heterogeneity is quite low. And then a fixed effects model would be perfectly fine to use in that setting. But invariably, that isn't the way the data is when you're performing a meta-analysis. So a random effects model is probably a more natural way to run your model. But generally the rule is, if your heterogeneity is under 50%, a fixed effects model is probably okay. If it's over 50%, you should use a random effects model. And if you're unsure, you should use a random effects model as well. Just to give you a little bit of background into that before we talk about forest plots, because I'll describe where you find the data in the forest plot now, and then we'll have a better kind of understanding as to what it actually means. So this will be very basic description, and for some, you probably have generated forest plots before, or you know how to interpret them. But the first thing to note is this is the data that you actually input into your forest plot. The name of the paper, and I usually use the year that the paper was published as well to differentiate it. And then, as we saw in the data tables, your mean, standard deviation, and the total number of patients that were included. And these were patients who underwent a robotic rectal cancer surgery compared to laparoscopic rectal cancer surgery. So these are your two groups, and this is the data that you've taken from your Excel table, actually put into the software package, and that has generated this forest plot for you. Here we see the weight of the data. And what this means is what proportion of the data within each study makes up the pooled analysis. And I didn't actually plan this, but this data is really evenly weighted. So you can see all of the data ranges between, comprises 12 to 15% of the overall pooled analysis. So this isn't a really good example to show the difference in weight, because the weight of the data will be represented by the size of the square on the right of the forest plot here. So you can see the small green squares. They're all the same size because the data is actually very evenly spread from the point of view of weight. And then the lines coming off each end of the square are the confidence intervals associated with the mean within each. So next we're talking about significance. So what we're trying to show here is our preoperative IPSS scores, significantly different between two groups of patients. And we can tell from the diamond shape here that they're not. And the reason that they're not is because it's actually crossing the midline. So for a finding to be significant, which we'll see in the next slide, that it needs to be fully separate from the central line. And the significance of the overall pooled analysis is represented in the test for overall effect that we can see here. So the p-value essentially for this pooled analysis is 0.12. And the heterogeneity for this analysis is 94%. So there's always a p-value associated with the heterogeneity as well, not to be mixed up with the p-value for the test of overall effect, which is actually the p-value for your pooled analysis. And because the heterogeneity within this forest plot is over 50%, we used a random effects model to analyze the data. Then when you're, writing out your results paragraph and you want to try and, put this information into, a written numerical format that you can include in your text. So this is what this forest plot tells us, and essentially to explain what the findings are. So this is the mean difference, the MD, because we're looking at the mean of an IPSS score between robotic patients and laparoscopic patients. It is a minus value because the reference we've used in the study is laparoscopy, meaning that in robotic patients that their IPSS scores are slightly lower. And we've also included the 95% confidence interval, which we have found here, so our mean difference, and then our p-value, which we found here. And this is what we actually include in our text at the end. So here is just a different format of the forest plot to just describe a few things that we didn't see in the last figures. So here we could see heterogeneity is equal to 2, so less than 50%, and we used a fixed effect model because of that. And this was a significant finding. So we can see the diamond, which is the graphical representation of the pooled effect, is separate from the midline, and the p-value for this study is 0.005. And this is how we write that from the point of view of inputting it into our results section. And just to show, we can see, because we couldn't see it in the previous figure, in this study, the weight is actually quite different. So the first study, for example, comprised over 40% of the entire pooled analysis. So the square representing the mean of the data is actually much bigger than, for example, in Park's study here, where it only represented 10%. So they're just some components of a forest plot to be comfortable with, to understand. And it's really important that you understand what all of this data means at the end as well. So this is your overall outcome, the 95% confidence interval of your outcome, and your p-value is from the test for the overall effect. So the previous examples looked at continuous data. Here is a study we did a few years ago looking at categorical data. And as you can see, this is why it's really important that you have your data tables correct at the beginning, because you can't see mean or standard deviation anywhere in these forest plots. Here we're looking at n values. So n values of the incidence of something happening and the n value of the total group. And other than that, it's the same essentially concept, because we're looking at categorical data, though we're not looking at a difference in mean value. So it's actually a risk ratio that we're going to generate at the end. And here we can see our risk ratio, and here our p-value is not significant. And again, the diamond is crossing the line. To mention very briefly, another element of data generation when we come to performing a meta-analysis is looking at publication bias, and for this you can generate funnel plots. These are usually when you generate your forest plot, you'll automatically generate a funnel plot for that finding as well. Technically, the funnel plot only needs to be generated for the primary outcome, but I think if you have any significant outcome, you should probably generate a funnel plot to include with that. And essentially, this is an expression of the study result over the precision of the study. And the precision of the study is usually the finding of the study divided by the standard error mean as well. What you want to have for if there's no publication bias is that you want to have each of the circle represents a study. All of the studies should be within the triangle and as evenly distributed around the mean average as possible. So in the previous examples, they were all related to pairwise comparisons. So comparing two different interventions, that would be the most common technique that you use or that you probably read about as well. But there are other meta-analysis techniques that I described at the beginning. So to briefly discuss them, diagnostic test accuracy, this is where you input the data from sensitivity, specificity outcomes from individual studies. The software package will analyze all of that and generate pool sensitivity and specificity outcomes, and also will generate area under the ROC curves, so you can examine the area under the curve for the pooled analysis of an investigation as well. Then I guess something that's probably becoming more commonplace now are network meta-analyses. So this is a really nice one comparing open laparoscopic, robotic and transanal TME approaches. So network meta-analyses are a little bit different to the ones that we previously described in that they will compare more than two outcomes. And this isn't something that you can do in RevMan, so you do have to use a different statistical package to do that. But essentially, the way in which the data is compared is that the comparison of treatments within a trial, so there's a head-to-head comparison, direct comparison performed with an indirect comparison of treatments against a common comparator within all of the trials that are included in the analysis. And this generates usually a hazard ratio and a 95% confidence interval. The only thing you have to consider when performing a network meta-analysis is you have to identify what is your gold standard or what is your reference technique. So in this study, for example, they use laparoscopic TME, and then your hazard ratio is generated as a percentage of that. Then the final technique that I'll discuss is looking at big data and omics. And I think this is going to be a really exciting area going forward. And this is an example of one that Helen and I did recently on metabolomic pathway, so a meta-analysis. But essentially, as research in the omics arena is increasing, we're getting multiple papers, multiple big data outcomes with regards to the clinical relevance of different elements of the omics pathway. And it's not only from a diagnostic point of view, but prognostic point of view as well. So essentially in this study, we wanted to look at, could we combine the relevant published data on metabolomics and how it may offer a prognostic benefit for cancer recurrence and cancer survival in colorectal cancer. And we included 9 different studies. Risk of bias is really important in this arena as well. And this is the Quadomics tool, which is an adaptation of the Quadus tool for diagnostic test accuracy that Matt previously described. And we were able to extract all of the metabolomic data from these papers, and using Metaanalysis, which is an online software suite that allows you to map metabolomic data to metabolite pathway information from multiple publicly available databases, and then generate your prognostic pathways at the end. So these are the kind of things that you can generate and you can look at. So this is looking at, what metabolomic pathways are active and upregulated around the time of surgery that can actually be prognostic when considering colorectal cancer recurrence. So the raw data from the papers was used, and it was mapped to these multiple pathway databases, and then the outcome is generated as you see here. The p-value in the on the left side is coded by the intensity of the red color and the size of the circle, and it's similar in the line graph as well. And the most interesting, I think, part of this is that you can actually generate crosstalk from genomic data to metabolomic data, or if you're working on the microbiome and transcriptomics, and you can really bring all of this data together. But furthermore, you can also, within the software, control for your false discovery rates and generate adjusted p-values. So it has really thought about a lot of the important scientific elements of what is required. So I think that was probably quite heavy going because there was a lot of data discussed and forest plots and, you know, heterogeneity. But I think the overall summary and take-home messages are the most important thing to do is to seek advice from a statistician and an experienced researcher from the very beginning. If you design your data extraction, data analysis plan properly, it will save you a lot of time and, you know, ensure the accuracy of your data and your overall outcomes. So plan. I can't emphasize that enough. Replan, you know, make sure that you have thought about everything, planned everything really clearly from the beginning. Then when it comes to the data itself, I think just selecting the correct methodology. So for example, if you want to, you know, compare diagnostic text accuracy or, you know, just basic pairwise or big data, just be sure that you have the methodology correct. And then decide on the variables that you want to include, and most importantly, the format of the data that is required, because that will save you a lot of time for having to go back and forth to papers again if you have the data incorrect. So thank you for listening. I hope that was of some help and informative, and look forward to taking some questions. Thank you.
Thank you very much, Christina, for a very informative and comprehensive talk on meta-analyses. We have a few minutes before the end of the webinar to take some questions, and some of them come through the chat box. I'll open up to both our speakers today. So one question which came through was, when is it important to make language restrictions when selecting studies for inclusion criteria?
So I think that the only reason you should exclude based on language, bear in mind that a systematic review is about getting the widest possible data set into your study, and just because it's not been published in, you know, English or a language that you have access to doesn't mean it's not useful. The issue is that if you don't have the resource available to get a reliable translation, because a rubbish translations, with a rubbish translation, you might as well not include the study. So for me, when I'm working on systematic reviews, I tend to only work with English papers that are published in English or with an English translation. Some journals will do publication of the English and local language, but because I typically don't have the resource to get a professional translation. That's why I set the limit. So it's up to you. If you are fluent in more languages and you're happy that you are fluent to a technical level and can read a scientific paper, then go ahead and include those. But if you can't, then consistency in saying we'll only limit it to a language is the way to go.
Great, thanks, Matt.
Christina, I might direct the next question to you, because I know you've had some experience with this topic. Are there any tips for systematic reviews of non-clinical topics, such as surgical education?
Yeah, so I think it comes down to the exact same basic principles that have been described tonight. And I think, sorry, I'll stop sharing my slides there. I think it comes down to just ensuring that your study design is correct, ensuring that your risk of bias has been adequately thought out and planned. And from the point of view of managing your data, it really doesn't matter that it is a non-clinical topic. You know, we did a study recently where we were looking at proficiency scores. So for example, EYSAC scores. So again, that's just looking at mean and standard deviations and total number of participants. So all of this is transferable across all domains, clinical and non-clinical, I think.
Great, thanks. I've got another question, which is a general question. What are systematic reviews and meta-analyses closely related? We see some papers in the title where both systematic reviews and meta-analyses are both used. Is there a difference between the two?
So a meta-analysis is just is essentially a way of saying pooling of data across a number of studies. And you shouldn't really be doing a meta-analysis without having been through a systematic process to identify the constituent papers, otherwise you end up with selection bias. So not all systematic reviews need a meta-analysis. You can do descriptive systematic reviews, narrative systematic reviews, but if you've done a meta-analysis, there better be a systematic review behind it. Yeah.
I would agree with that point. And I think something else to consider as well is sometimes you'll find that, you'll get really important clinical information after a systematic review, but only a proportion of the papers are actually comparable enough to be able to meta-analyze the data. So for example, if you're looking at, as one of the examples I use in the talk, sexual function after rectal cancer surgery, you had lots of comparable studies in men, but none in women. They were all very different. So, you can still report that in a systematic way, but then, quantitatively meta-analyze the male-based data. So.
There's a question on the chat directed to the panelists that Matt, you might be in the best position to answer. It's asking about experience with the NICE HDAS.
The HDAS question, yeah.
Might explain for those of us that are maybe from an international audience and what the what it is and whether it's of use.
So NICE is one of our government bodies that is tells us what interventions are cost effective, and they have a big strand in evidence assessment. One of the tools that they provide access to is this healthcare database advanced search, which is kind of an Uber search engine that I believe gives you access to most of the search engines I've talked about. It does so through sort of a more straightforward syntax than is used with some of the other search engines, and without necessarily the things like the commands that will search only titles and abstracts or things that will use multi-parameters. Its search is a bit different. You can use it as long as it's a way of searching a number of databases. You may not know which papers come from which database, and you may lose some of that functionality of the search. So, I mean, I would say it's down to personal preference. My gut feeling would be I would like to search each database through a search engine that is designed for it. But if this is what you've got access to, it's what you've got access to, and you can use it. You just have to justify it to a peer reviewer at the end.
Great, thanks. And now on some more statistical questions from another viewer. They say that, and they ask if some authors present an odds ratio or a hazard ratio following univariate analysis, while others provide a value following a multivariable regression, can you use both in your analysis and your results?
So you can't use both in your analysis because whatever you use, you just have to use it consistently. So, but there are formulas for converting your data into the variable that you want to use. And the link that I used on the conversion slide, it has all of those equations on it. So I would the most important thing to do is to think about what are you trying to show? Is an odds ratio or relative risk more important for that? And then, I would think if you're not sure with regards to the data coming from the paper as an odds ratio, relative risk, then I would contact the corresponding author and ask them, because I think that's probably the most accurate way to do it. Because it's not just the odds ratio, the 95% confidence interval, the standard deviation, it all has a knock-on effect. So the purest way would be to contact the corresponding author. But there are some really good ways of manipulating the data as well. Matt might not agree, he has his hand up.
No, actually, I'd interpreted the question slightly differently. I think if you're looking at a study and they've done a model where they've done a univariate model, so looked at the impact of each item individually, and then done a multivariable model, and only some things have made it to the multivariable model, so predicting outcomes or whatever. I think that you should only pool if you're pooling odds or whatever, then you should only pool multivariables with multivariables and univariables with univariables. You shouldn't really mix them because they've not been risk adjusted in the same way. And we can get into it. There's a whole other debate, I think, about pooling of models and that kind of adjustment. But for the questioner, I think that's what I'd say.
I agree with all of that as well. There was a question actually on the number of studies that are required with regards to, so the general rule is you should have at least 4 studies from a statistical point of view. Now, there are publications where there's less than that, but as a general minimum acceptable standard, it's 4, but really greater than, kind of probably 6 is the middle of the cutoff point for that, I would say.
I think as well, just on that point, there's another thing just to mention about not just the number of studies, but the size of studies. If you're going to pool 3 studies, 4 studies, and they're all really tiny studies, you're going to get such wide confidence intervals. You can get confidence intervals you could drive a bus through. So I would, if you're going to pool data from a small number of studies, make sure they are decent sized studies that will give you a precise estimate. Otherwise, you might as well just guess.
There's a question on the chat from Ellie Watson. Could you please discuss how to approach assessing the quality of included evidence / the application of the grade tool? And a second question about how to assess for meta biases, particularly publication bias. Matt first, maybe. Then Christina.
Yeah, thanks for this. This is a really good question. So grade isn't a tool that I preferably use. I think grade is, when I'm doing sort of bias assessments, I tend to like the tools that get me into the nitty gritty of how do we select patients, whatever else. I think grade is very good for giving you an idea about the, the summary tool is very good for giving you the sort of accuracy of estimates and how certain you can be, and gives you ideas of numbers needed to treat and where those might lie. I think particularly for interventional studies, that's quite useful. For the studies that I've tended to do, we've not gone down that territory so much. There are online tools that let you establish a grade. I think there's a browser plugin and that you certainly downloadable tools that you basically populate with the data after you've done everything else. So I would use grade as an add-on to other things, and there's decent enough online walkthroughs that should be able to take you through that. So it's something plus grade is how I do it. Meta bias is particularly publication bias. Well, I think that's a good one, and it's often done statistically because we do get into sort of meta-analysis territory, and Christina alluded to things like publication bias. It's difficult to answer that question without a little bit more stuff there, and it's an interesting thread to pull on. There are the risks of positive, negative studies, and the risks of publication there. Some of that really you have to draw out a narrative. I don't know. I'm not aware of any tools off the top of my head that will get you a nice number or whatever at the end to talk through.
I also think the funnel plot is really misleading, and I don't particularly like it because it only can tell you what it knows, and the whole concept of publication bias is the unknown. So I really just like funnel plots, and I generally just put them in as an appendix or supplementary data because sometimes you're asked for them and you have them, but I really don't think that they add anything, because as you say, it's just a statistical outcome of what you've told the analysis, and it's actually what you don't tell the analysis or can tell the analysis gives you way more information about publication bias. But I think it's a really difficult question to answer. I don't know.
I've got to say, I never thought I'd hear someone say in all earnesty, I just don't like funnel plots. So, yeah. Thank you for that.
You're welcome.
There's one last question on the chat that we might just address before we wrap up. And that was, what is the magic number, Matt, maybe, of databases? I know we all go through the rigmarole of going through a certain number of databases, but really there's a lot of overlap between them. What is the magic number, and is that really based on what?
So there is some meta research on how many databases you should look into, and that you do hit a point of diminishing returns. I think if you are a researcher in surgery or surgical oncology, I would expect you to have searched Medline through an appropriate search engine, Embase, and probably Central as well, plus a hand search or a grey literature search. So in keeping with 90s hits, 3 is probably a magic number. A bit more is nice, but 3 is a magic number.
Anything on that, Christina?
I think I would agree. I think that's pretty reasonable.
So with that, I'd like to give a big thank you to our speakers for a really informative session. Thank you to my co-host Trevor Young for joining us at a very early hour of the morning here in Melbourne. And I'll hand over to Andreas just to wrap up. Yeah, he's been very supportive of the webinar.
Yeah, thank you so much. Thank you to the speakers. Thank you to the wonderful chairs. I think we learned a lot today as a part of the EYSAC Research Academy. And I would like also to thank Anna Galan from ESOSTAF, who made it possible for this European-Australian collaboration webinar today. So it is already quite late. Thank you again for hosting us. And I would like just to highlight and invite you to our next webinar, which is actually in a little bit less than 2 weeks, on the 2nd of May. We will have innovations in surgery as our topic, and Declan Soden will present about innovations in cancer therapy, electrochemotherapy, and immunotherapy. So stay tuned, keep in contact with us online, social media, email, and whatsoever, and have a wonderful evening or day wherever you are. Thank you so much.
Thank you.
Bye.
Bye, nice evening.